Hi, Dr. David Fisher. How are you?
I'm good, thanks. Good to see you, Steve.
Well, thank you so much for taking the time to join us on this Beating Melanoma project. And to all the viewers and the readers out there, we have a great treat. Dr. David Fisher is actually the Edward Wigglesworth Professor of Dermatology, Biological Chemistry, and Molecular Pharmacology at Harvard Medical School. And he's also the Chief of Dermatology at the Mass General Hospital. And he's a Director of Melanoma Program at Mass General. We have a real, truly expert, and they're going to shed some light and talking to us about melanoma and some of the skin biology. So welcome again.
Thank you. Good to be here.
Now, before we jump right into some of the latest research you and your group is doing on melanoma, maybe you can shed some light. Why is it that some patients have higher risk for developing melanoma, right? Especially people with lots of freckles, red hair, and why is that?
So it clearly is the case that the biology of skin is contributing to the risk, but it is not the only component to it. There is an interaction between the skin, particularly the pigmentation in the skin, and the environment, where we are particularly focused on the role of ultraviolet radiation. And within the ultraviolet region of the spectrum, there are different components, and particularly UVB and UVA appear to be contributing to that risk. And we think there are interactions between melanins. Certain melanins can have protective effects. The dark pigments can actually have protective effects, whereas the light pigments, the red blonde pigments, actually seem to worsen the risk. They seem to induce a chemical injury in the skin, particularly in combination with certain wavelengths.
Before you go on a little bit further, maybe just to clarify, since we have a lot of general public out there, what is melanin, what is pigment, and what kind of cell makes it, and why what is those things?
Sure. So melanin pigment is the material that causes the pigmentation, the color that we see in our skin or in our hair. It's actually the same pigment in our hair that would be present in our skin. So given the wide variety of hair colors that you see out there on the street, that type of variety actually exists within our skin, but it is not as apparent, actually. The variety of skin pigmentation, while there's a wide range, it's not quite as wide as what we can see in the hair, because in the hair, it's much more concentrated than it is in the skin. It's more dilute. So for example, my skin is very light. My hair is very dark. My skin is probably pigmented quite similarly to the skin of a red-haired person, even though the pigment in my hair is not, primarily red or blonde at all. It's quite dark, and so the pigmentation components are actually nonetheless very important in controlling or regulating, modifying the risk of melanoma skin cancer, as well as non-melanoma forms, many the most common non-melanoma forms of skin cancer. And so that is a very important area that I would say, stepping back and looking, we would say it's an interaction between your genetics, what you're born with in terms of skin biology, including pigment, and what the environment is inflicting on your skin, part of which is determined by how you choose to expose yourself to that environment. One thing you did ask, I forgot to answer, is what kind of a chemical is pigment, melanin? It's not a protein or a lipid or a sugar. It's actually what we would call a small molecule. It's metabolized from an amino acid, which is a building block of proteins. It's the amino acid tyrosine, and it gets modified chemically, and then later on it actually does aggregate and form these large polymers that absorb light, and that's the reason that we perceive them as having color, because they absorb in certain wavelengths more than in other wavelengths. But that's a super quick summary of what melanin is. And its composition has a lot to do with what our risk is. People with dark pigmentation in their skin have dramatically lower risk of melanoma in the sun-exposed parts of their skin. than people who have light skin. And also the ability to tan is a very important variable. People who don't tan or tan with great difficulty or burn very easily are at much higher risk of having melanoma in the sun-exposed parts of their skin. In the non-sun exposed parts of the skin, like your palms and soles, or in inside of the mouth, or in the genital urinary areas, those forms of melanoma occur at approximately a similar rate in people with light or dark skin pigmentation.
I think I interrupted you before. We talked about melanin is this pigmentation that's produced by melanocytes, right? And you mentioned there you use some technical name eumelanin, which is representing the darker pigment, and the pheomelanin, that is the lighter pigment, right? And you were in the process of talking about when those melanin, the different type of melanin, interacting with the UV radiation, something different biologically happened, right? And that causes one group to have higher risk. Maybe you can continue with that thread.
Sure, exactly. So actually, there were studies way back in the 1960s showing that red blonde pigments, the so-called pheomelanins, actually on their own have a chemical reactivity that produces what we call oxidative stress or reactive oxygen. This is the opposite of an antioxidant. Antioxidants we think are good in being protective. These are pro-oxidant. And even more so, if you shine UV radiation on the red blonde pigments, the light pigments, they emit even more pro-oxidant stress. In contrast to that, the brown black pigments actually are very potent antioxidants. And a component of their protection against sunlight is probably that protection against the oxidative risk that UV ultraviolet radiation is inflicting. They have some ability to actually absorb UV itself, like a sunblock, but it's surprisingly weak. It's not a very strong SPF. The SPF, even of pure dark pigment, is single digit SPF. It's not like these SPF 30, 50, 90, anything like that. So the protection against melanoma, the likelihood of getting melanoma from within sun-exposed skin in people with darkly pigmented skin is hundreds-fold lower. And so there is a benefit, we think, even from this antioxidant effect that is probably beneficial and protective against melanoma risk.
So another question is, so we got some basic understanding about melanin in our different group have different colors. And why do people get tanned? And what are the signals that triggers that?
Yeah, so really important question. When we say why do people get tanned, I mean, part of it is like, how does it actually happen?
Yeah,
and
exactly.
There's another question is, why would people actually do this? And in evolution, in the last 10,000 years of human evolution, like where did this come from? So a lot of questions there. It's a very, very important question, and I'm glad you asked it. So we know that the pathway, which is initiated by sunlight, by ultraviolet radiation hitting our skin, and that is ending with the production of melanin, which is the tan, is actually, it's a DNA damage response. It's an injury response. It's an injury response whose output is the production of dark pigment. It's probably not a pathway that evolved for purposes of beauty. We don't know this for a fact. It's very difficult to explain evolution. But it does seem to have evolved in more likelihood to be somewhat protective against the possibility someone would go out in the sun tomorrow or the next day or the next day, because it can help protect against sunburning and really injuring the skin if you live in a very sunny area. And so we think that it offers protection and a benefit by allowing the skin to adapt to the intensity of ultraviolet radiation, because ultraviolet radiation injures the cells of our skin. It damages the DNA, it mutates the DNA, and those are the kinds of events that have multiple outputs. One is a risk of cancer. Another, frankly, much more common than that, is the what we call photoaging. It causes you to look older. It doesn't do it the next day or the day after that, but it produces everything from wrinkles and atrophy to changes in pigmentation that will last, that sort of dark, leathery skin, from some people who've been in the sun enormous amounts of time. By the time they're 40 or 50 years old, you can just look at them and you can say, I think this person was a tanner, really, really spent a lot of time in the sun. And so that's part of what is going on. When we think about the evolution of skin pigmentation, we remember that there is one biochemical pathway that ultraviolet radiation contributes to that in evolution many years ago was one of the only ways to get the vitamin D, one of the critical synthetic steps in making this molecule. This is actually an essential vitamin. Our bodies need vitamin D. And back in the day when we couldn't go to the local pharmacy and get a pill that was cheap and effective to replace vitamin D if you didn't have it, sunlight actually was contributing that chemical reaction for that one critical step. And so for populations, our current understanding is that for populations living at very high latitudes where the intensity of ultraviolet radiation for much of the year, let's say in Scandinavia, is very weak, very, very little UV radiation, very little sunlight, the days where there may be minimal sunlight, days on end, those people 10,000 years ago may have very serious vitamin D deficiency. And if you look at the skin of people whose ancestors evolved to live in those areas, they have light skin and they typically don't tan very well. And that has led to the hypothesis that this was protecting every last ray of UV radiation to contribute to vitamin D production. Even if they were getting some risk of skin cancer, and they were, because these are the people who are at risk of skin cancer, it was a little bit lower because the intensity of ultraviolet radiation wasn't that high in those places. And the skin cancers that formed, even if they were fatal, and melanoma unfortunately can be fatal, they tended to be fatal after the reproductive ages of these people. So getting through childhood, when vitamin D deficiency would be fatal if you don't have it, was actually more important if you had to choose the bad outcome. If you could at least make it through vitamin D deficiency 10,000 years ago, you may die of skin cancer in your 30s or 40s, but at least you made it through childhood. Now, things have changed, of course, because we have other ways of getting vitamin D that for populations that need it, you can take a supplement which is virtually identical in its efficacy in replacing vitamin D. And on the other hand, the risk of UV has gone up because many of those very light-skinned people have moved away from Scandinavia or high latitude locations, and now they live in, let's say, Florida or
Australia, right?
Or Australia.
Perfect
exactly.
experiment, yeah.
Correct, exactly. And so these kinds of discrepancies in light skin, dark skin, and UV radiation intensity have led to pockets of very, very severe skin cancer and melanoma outbreaks as well.
So just to summarize, I think we touched a lot, right? We talked about what melanin is, what pigment, the melanocyte makes it. That's the same cell that can develop into melanomas. And we touched a little bit about the kind of evolution of vitamin D. And so to just summarize for everyone out there, so basically, in essence, due to evolutionary forces, that as human race migrates from lower altitude, let's say, such as Africa, and all the way to Norwegian, Ireland territories, the selective pressure favors to have individuals with a pheomelanin, lighter skin color, because you need more of the vitamin D production to enhance bone strength and musculoskeletal, right? And then and so that is the cause. But you also touched a little bit about the difference between eumelanin and pheomelanin. Eumelanin is the people with a darker skin. Those have more of a pro-oxidative antioxidant, so they protect the skin from UV radiation. In contrast, the lighter skin, the pheomelanins, they actually not very effective. At the same time, upon UV radiation that causes DNA damage, that triggers the cellular machinery to produce melanin to protect the skin. And those pheomelanin is generated actually creates free radicals, oxidative damage that further creates more damage. So you get into this vicious cycle in essence.
Yes.
Yes. No, I think this is a really, really interesting thing because, most people think photo protection using sunscreen is a very just kind of a straightforward thing, right? But there's so much evolution, so much and nuance behind it. And also, I don't know if I have time, but I know in the same molecular pathway that generates this melanin production, that's also creates endorphin. And that is also another reason why some people can be addicted. That's why some people, when they stay on the sun, they feel really good about it.
Exactly.
We're going to shift gears a little bit because I'm dying to sort of ask you a lot more questions about melanoma. And so, you're considered as you are actually one of the world experts when it comes to melanoma. So, what, in your opinion, what are some of the major breakthrough in the last 5 years, I would say?
Yeah. So probably, and now I think we're fast forwarding to actually the therapy of melanoma rather than the prevention or the
Sure.
formation of melanoma.
Sure.
So in the therapy of melanoma has been, I would say, one of the most striking stories, I would say, actually in the history of oncology, in the history certainly of modern oncology. It's probably as important as it was when people first started doing surgery altogether to be able to remove any kind of a cancer. What has happened is that we have therapies now, primarily immunotherapy, which are able to generate long-term disease control, and I think it's pretty fair to call it a cure. In a significant fraction, it's still unfortunately a minority, but in a significant fraction of patients who have metastatic advanced melanoma, where despite the fact that this disease has spread to distant organs in the body, and in some cases it can be a heavy load of that tumor, that these drugs can activate the immune system to destroy the tumor and generate what looks to us like a cure. And we're at a point where it's probably successful in the range of a third to a half of patients who have advanced melanoma. Until recently, the ability of drugs to cure advanced cancer has been limited to a tiny, tiny fraction of cancers. And in those instances, they're usually chemotherapy drugs, which, while they work well in, let's say, Hodgkin's disease, testicular cancer, pediatric leukemia, certain very specific scenarios, what has happened with melanoma is an extremely deliberate, mechanistic, basic science-driven process of understanding the pathways that the immune system uses to regulate itself, and then not just make a drug of sort of something that chew on a plant leaf and it happens to make a tumor shrink, although we'll take that if it happens, but these are actually engineered molecules that are able to activate in a designed fashion, activate the immune system and cause it to recognize the tumors and kill them. The success in that is more than 5 years now. It's been closer to 10 years, yep. and that's been fantastic. In the past year, and the progress is continuing. And I'll tell you what I think is going on right now.
Yeah, that's what I'm really curious about. Yeah.
Yes. So along those lines, there are now three, I should say, classes of drugs activating different, what we call immune checkpoints, in order to, they're not activating the checkpoints, they're actually inhibiting the checkpoint to release the immune system to become active. The checkpoints exist in an inhibitory fashion, and by blocking the checkpoints, you have a double negative, you activate the immune system. And in allowing the immune system to become activated, it can kill the tumor. The first of those drugs had a modest activity, but a real one. The second of those, which was a class of drugs that targets the PD-1 checkpoint, has a huge amount of activity. And that was the lion's share of that 1/3 to a half of patients. And then most recently, there's another checkpoint that has had, again, a very modest impact, but it is now FDA approved, and it is just rolling out now, as probably will be in a combination approach, several drugs to try to really boost the ability of immune therapy to work. It's probably closer now to 50% of these patients having that activity.
So
That's
I think
one.
so
Yeah.
that everyone knows, the first one is the CTLA-4 inhibitor, the PD-1, and then you're talking about the LAG-3 inhibitor. So those are the three. And so Olive, just curious, why do you think, how do we know at this point? Like, can we even predict which patient will respond? Is there any kind of assay, or is it something that your lab is working on to study the immune microenvironment?
Yeah, tremendously important question. And it's important for a reason that I'm about to talk about with the other big advance that's been happening, and I'll get to that in just a second. So my lab is interested in this, but so are dozens of others, because it's such a screamingly important question. One of the reasons it is so important to try and predict which patients will have a good response and which will not have a good response is that, like many other drugs, these drugs can have side effects. And so, on one hand, if the drugs did not have side effects, everybody should just try them. I mean, what do you have to lose other than time, which of course is significant as well? But if they have side effects, and if on occasion those can be serious side effects, then you don't want to be giving a drug that could not work to a patient who may then have toxicity from it. And so that's why we tried to understand this. So a lot has been learned, but not everything. So what do we know? We know that tumors, which prior to the initiation of therapy, before you start treatment, when you look under the microscope at the tumor, you can see there is evidence of an immune infiltration. There are already cells of the immune system at the tumor before treatment even started. That is a very good predictor. It's not, unfortunately, a perfect predictor, but it's so strongly suggestive that this is optimistic that these drugs are going to work. There are other predictors that tend to go with this. For example, when you look for mutations in the DNA of a tumor, melanoma is one of the cancers that has among the highest density of mutations. And we can actually see from the chemical nature of those mutations that most of those were caused by ultraviolet radiation. There is a distinctive chemical feature, we call it a signature, a chemical signature of ultraviolet radiation. And that probably is part of why the immune system can recognize the tumor, part of why there are immune cells sometimes already in the tumor before any treatment even happened, because these mutations cause some of the proteins to be altered in the melanoma. And the immune system has, in all of us, has been tolerized to our normal proteins. Our immune systems should not attack our normal proteins. But if those proteins have been altered a little bit, the immune system gets confused, sort of like, is this COVID? Is this influenza? Is this some foreign fungus or bacteria? What is this protein? And those mutations may actually have one good feature. Unfortunately, they contribute to the cancer risk in the first place, but they also can allow the immune system to recognize the tumor. And if you get these drugs, these checkpoint inhibitors that liberate the immune system to act on that recognition, it can actually free the immune system to attack those mutated proteins and actually kill the cells. And that is a model of what many people think is going on with this successful immunotherapy. There's no simple algorithm, though, for knowing which mutations predict who will respond to these drugs. Exactly how many immune cells you need in the tumor to predict with great specificity. But people are working very hard to try and understand what are the risk factors so that we'll have better models to predict that.
I know there's a number of clinical trials try to combine, let's say, radiation and try to combine different things. The idea is to release some of the antigen and then adding the immunotherapies. But is there anything that you guys are doing in the lab that biologically engineering or boosting this immune efficacy?
Yeah, so there actually are things that we're doing and other labs as well, which is to try and artificially inflame a tumor. There actually are certain drugs that that evoke inflammation. If you think about a vaccine, that's what it does. When you're giving if a person gets a COVID vaccine, for example, they're getting an injection that has a key protein, the spike protein from COVID. And then they're also getting things with it that trigger the immune system to become inflamed, to be attracted to where that COVID protein is, so that it will stimulate an immune response. And so the world of vaccines and the world of other pro-inflammatory strategies, we think can be utilized to try and further activate the immune response. And if that worked, in principle, it would not be limited to melanoma, though we can certainly use more help in melanoma, but it might work in other cancers where the immune system is not sufficient currently to really produce these kinds of responses.
No, those are really great. So, I mean, I know we're kind of running out of time. You and I can chat about this for another hour or so. One of the things is, you are in the forefront and you're not only seeing patients, but you're also doing research. What can you tell patients at this stage who have, early stage of melanoma versus someone who has advanced stage of melanoma? Because everything we say, there's an enormous power of healing, I think, right?
Yes.
What kind of pearls of wisdom do you have?
So I think the striking success that therapies have seen in melanoma is actually marching on. There is even more, and even though we're almost out of time, I'll quickly try to summarize. One aspect that I think is one of the most exciting, very recent developments within the last 1 to 2 years, and that is taking these treatments that were given to patients with metastatic melanoma and moving them up to earlier stages in the development of melanoma. So when it's still confined to the skin, but where we know, based on its thickness or based on local spread to lymph nodes, that these people have a statistically elevated risk that even after it's been fully removed by surgery, they could have a relapse. And unfortunately, that is the case for a significant fraction of patients with melanoma at earlier stages. And so some recent trials have tested those very same drugs now in the earlier phases and seen significant benefit, major benefit, as measured by the lower likelihood of relapsing within a couple of years afterwards. Unfortunately, these data are so recent that we don't know whether these are the same patients who, if they did relapse, would have been cured by those drugs at the advanced stage. And that's a terribly important question. We need to learn the answer to that question, because there's an uncertainty about whether to give these drugs to patients with early stage 2 or stage 3 melanoma, because the majority of those patients are never going to relapse after the surgery is done. So once again, we have this predicament. We could give a drug that may lower the risk of relapse, but many of those patients are at risk of side effects. And so if you didn't need it, if that tumor would never have relapsed anyway, you would certainly not want to expose those patients to that risk. So again, as you asked before, how can we tell which patients are going to benefit and which not? Research is terribly important to try and figure that out. What features will predict the patients at greatest risk of having a relapse and potentially responding to these drugs, so that we can really appropriately tailor the therapy for the patients who will benefit from it the most. But coming back to your bigger question, I think, which is for the patients who have melanoma right now, family members or patients who are at risk of having melanoma, many people would say in the history of the disease, this is the time to get it if, God forbid, you need to get it because we have
Agree.
greater tools than we ever have had before. And the research engine is running, and it is running at 100 miles an hour. There are discoveries constantly coming out. There are improvements constantly coming out. So for myself, I think I would speak for many of my colleagues, this is a very optimistic time in the field. We're not where we need to be. There's much work to be done. But we are learning so much. And again, because it is based on the science and the mechanism of the disease, we really think we're getting it in the corner here. It's stuck. And we don't have yet all the tools we need, but we know the questions better and better. And hopefully, as those tools come together, the therapies will really reach where we think they have to be at the end.
No, that's a really optimistic outlook, right? And I completely echo that. And thank you again for your time. And we touched a lot, and we went from evolution, talk about prevention, and moving on to the future of the current status of melanoma treatment, and moving on towards the future. Seems to me it's a more of a personalized approach, and if we can better predict who will develop those progression, and how do we figure out which patient will benefit from the immunotherapy, that you can sort of maximize the therapeutic benefit while reducing potential side effects. Well, David, thank you so much. I think hopefully I can invite you back next time, and maybe next time I will invite you, and you have to come on board and play your cello as well. Thanks again for your time.
My pleasure. Good to see you. Take care.
Likewise. Bye-bye.
Bye-bye.